Amino acids are the building blocks of proteins. They are protein monomers. The structure of an amino acid is made up of a carbon, a carboxylic acid, and an amine functional group with a side chain specific to each amino acid. Here is a representation of an amino acid that shows that there's an amine group, a variable R side chain, an alpha carbon, a carboxylic functional group. The variable R chain is different for each amino acid, and it sort of determines the function of the amino acid. Amino acids are coded by a sequence of 3 nucleotide bases. It is a triplet code. This triplet code is determined by the genetic code that gives instructions for the construction of a polypeptide or protein. It is a degenerate code. All amino acids except methionine have more than one code. The triplet code that codes for methionine is adenine, uracil, and guanine. Amino acids are linked together by peptide bonds. They react to form a dipeptide and polypeptides by a condensation reaction. Condensation reaction is where a small molecule is lost to form a macromolecule. There are only 20 amino acids used for protein synthesis. These amino acids can be classified as polar, nonpolar, and electrically charged. Due to the side chain group type, they can also be aliphatic, acyclic, aromatic, containing hydroxyl or sulfur. An example of an amino acid with a sulfur side chain is cysteine.
And this one is alanine, and the 3-letter abbreviation for this one is ALA. The 1-letter abbreviation is A. And this is the simplest out of all of the amino acids, seeing as all it has is a methyl group. The second amino acid that I'm showing you is cytosine. The 3-letter abbreviation for this is CYS, and the 1-letter abbreviation for this is C. And it has the amino group and the carboxylic group and the alpha and the beta carbon, like I explained before. But the only difference with this is that it has an extra carbon and a sulfhydro group. The next is asparagine, and the 3-letter abbreviation for this is ASN, and the 1-letter abbreviation for this is N. And it has the same as I explained before. However, this one has a carbamide group, which basically just consists of an extra carbon, main carbon here, the double bond O, and the NH2. This one is arginine, and the 3-letter abbreviation for this is ARG, and the 1-letter abbreviation for this is R. The difference with this one is that it has a guanidinium group, which is that right here, and it has an extra 1, 2, 3. So it has the 2 carbons, then it has the guanidinium group right on top of that. And last but not least, we have aspartic acid. The 3-letter abbreviation for this one is ASP, and the 1-letter abbreviation, however, is D, which is quite different to all of them.
Right, this here is the structure of glutamine. Glutamine can be abbreviated to 1 letter of Q. As you can see, it has 2 double bond oxygen on either side of the chain. It has a carboxylic acid group here. It has 2 amino groups, one here and one here. And the group, this part of the chain is the amide linkage of H2N and double bond O. And yeah, this is the structure of glutamine. This is glutamic acid, as you can see from the chemical formula and structure. You can see there's 2 carboxylic acid groups attached to either side of the chain. There's 1, 2, 3, 4, 5, 6 carbon and 6 carbon backbone. Glutamic acid can also be simplified into a single letter of E. As a result of the 2 carboxylic acid on either side of the change, it has a pKa value of 4.0. This is glycine. This is the chemical structure of glycine, and the side change is replaced by a single hydrogen atom, and as well as that you can see the amino group of H2N as well as the carboxylic acid group of C double bond OH. Glycine can also be simplified to GLN. Next one up is histidine. As you can see, the chemical formula and structure. It has a meso group which consists of 2 nitrogen atoms and 3 hydrogen atoms. This is all is connected to the main backbone of the amino acid. As you can see, the NH2 and the C double bond OH. And histidine has a pKa value of 6.0. Next one up is isoleucine. As you can tell, this is the chemical structure of the amino acid. It has 2 chiral carbon atoms, which means that each carbon atom, this one here and this one here, are attached to 4 different groups.
So this is leucine. This is an amino acid with the alpha taric group. It has the 3-letter abbreviation of LEU and the single letter abbreviation of L. This helps you remember through this red area here, as this kind of forms an L, and the L is with the beta carbon here, and then with a methyl group, with then another methyl group making the amino acid, the leucine, with the backbone. Lysine is with the basic group. It has a 3-letter abbreviation of LYS and a single letter of K. This helps you remember, as this is the kind of start of the K, but without the back. And if you just do this area, there's the 2 carbons, join those to the base carbon here, and through that carbon here, add an amine group, and that forms the amino acid along with the backbone. Then there's methionine, has a 3-letter abbreviation of MET and the single letter of M. This is a sulfur containing group. This is the sulfur here, and the M is kind of formed with the sulfur in the middle, forming here in the red, and if you draw on that to the base carbon, it kind of forms the amino acid on the backbone here with the sulfur in the middle.
So here we have proline. Its 3-letter abbreviation is PRO or pro, and its single letter abbreviation is P. It's got a 5-membered ring over here, and it's a cyclic molecule completing itself on the NH3. If you think of it like a P over here, then you have proline. Here we have phenylalanine. Its 3-letter abbreviation is PAG, single letter F. We have a phenyl group attached to the backbone. We have A with the benzene ring connected to the backbone. The carbon there, the beta carbon.
Valine is an aliphatic compound with the standard amino acid base with a beta carbon extending from the second carbon, and from this extends 2 methyl groups in a V shape, which coincidentally is the same letter that valine abbreviates to. Threonine is almost identical, except it has a hydroxyl group here instead of a methyl group. Serine is kind of different because it only has 1 group extending from the beta carbon, which happens to be a hydroxyl group. Serine and threonine are both hydroxide compounds. Tryosine. This one is different because it has a phenyl ring here extending off of the beta carbon, and it also has a hydroxide extending from the last carbon of the phenyl ring. Tryptophan happens to be most complex. This one has a phenyl ring here on the end, and it also has a 5-unit ring here which has a nitrogen in it and which connects to the rest of the chain. The easiest way to remember this is by drawing out the regular amino acid base, drawing a W, so this is your W, and from this you can draw your 5-point molecule and your phenol. Thank you for listening.